Okay, so we are on. Thank you so much, everybody. It's such an honor to see a lot of old friends here again, and I'm glad to see our new colleagues to this meeting today. It's such an honor to have you welcome to INC's annual meeting or scientific meeting or breakthrough breakout sessions, whatever you want to call them. But the purpose is that we wanted to gather all of our people, stakeholders who are a part of INC for years, back again together so we can interact with them, meet with them, discuss the work that we've been doing at INC for many, many years. One of the things I wanted to say is that this is 10th year of INC's establishment. I think we started in 2015. So yeah, we've been doing this for quite a while, and I'm really honored to be a part of to be able to be a part of some of what we've been doing over the past decade. So just a little bit of a house rules. We're going to have frequent morning breaks, frequent breaks. Lunchtime is going to be at 12:15, and then there's another break. We are going to be recording this meeting, and the recordings will be made available after the event. And I think there's going to be a photographer and videographer from CPATH who might be taking random pictures. Restrooms are outside on the left, and I think it'll be good if we could silence all the cell phones so we can actually listen to the sessions that are going to be there for all of the day to day. Again, my real honor to welcome you. I'm actually going to invite one of my colleagues, friends, and mentors, John Davis, who has been a part of International Neonatal Consortium since the day INC started. He has seen through all good things, okay things, bad things in neonatal drug development. Hopefully, most of them good. But he wanted to provide some opening remarks, talking about challenges, opportunities for developing drugs for this very fragile population that we all really care about. So I'm going to invite Dr. John Davis to the podium so he can talk about his experiences.
Thanks very much, Kanwalji, and thanks to you and Klaus and Colin and others at CPATH. Especially my thanks to Christine Barry, who's our project manager, who makes this all work every day. And it's really a pleasure to be here on our 10th anniversary. It's something very special, and I think we're all delighted to be here together. It was wonderful seeing old friends and being in the same room together after many years. So I wanted to just get the meeting kicked off by putting in perspective some of the issues with neonatal intensive care. 6% of the now 3.7 million births each year end up in the NICU. We have 100 more admissions at Tufts than we did last year. This year and last year, we were 100 admissions ahead of the year previously. So it was like a 25% increase in 2 years. So the amount of business we're seeing is really significant. The prematurity rates in the US of 11% are the highest of any affluent country in the world. It's fascinating if you come from a country in Europe or Japan or others as a woman and you have very low rates of prematurity in your country, when you come to the US within 4 years, you're as bad as anybody else. So whether it's environmental, toxic, or other exposures, we can't seem to figure out, despite millions of dollars spent on genetic studies and others, why that's the case. We also have the highest maternal mortality, perinatal mortality, and infant mortality of any affluent country. And the total cost of prematurity is over $29 billion a year. And yet, there's really been marginal improvements in survival and outcome in the last 20 years. And 80, some people say as high as 90% of all the drugs we use in the NICU in the US and in Europe, when Carol and I talk about this, are not formally FDA or EMA approved, and suggesting that there isn't sufficient safety and efficacy data to support the product approval. And the last drug was with a group of people that I worked with to invent pulmonary surfactant in the late 80s and early 90s. And that drug, which was approved in 1991, was really the last drug in premature babies that had really significantly improved survival and outcome. So it's been well over 30 years since we've had another drug. We did have caffeine approved, but I used to work with jacaranda, and we used to take caffeine and grind it up and mort it in a pestle and put it in a solution and give it to the babies. So that was interesting times. So why is it so difficult? Why do we need this organization more than ever? Well, it's a small market, rare disease, and as we know, very, very high risk and liability. In the courts now, it is not uncommon for damaged baby cases to go over $200 million awards for one baby who's been injured during labor and delivery. What we also know is there's few relevant preclinical, clinical, and digital models. There's difficulty with study design and outcome measures. And as many of us talk about all the time, the need to wait to determine outcomes. And that waiting is complicated. For companies who are in the field who want product approval relatively quickly, well, if you're waiting for a Bailey exam at 2 years, and many of us say, well, that gives you gross motor and language things, but you really need to wait to 5 years to look at the behavioral, social outcomes, visual outcomes. How do we expect anyone to do that? There's lack of quality real world data to generate real world evidence, and we're working very hard on that. But it's really the national and international efforts like this one with legislative efforts, regulations that are help driving innovation in this area. So part of it is neonates often have many rare diseases. So now there's improved diagnostics to identify rare diseases. In our Gemini trial, where we just took 400 babies from throughout the United States who were sick, that was the only entry criteria for the trial, had to be sick. It could be seizures, it could be respiratory disease, it could be anything. And we found a genetic variant in over 50% of the babies that helped explain the phenotype. The in utero diagnosis with ultrasound, MRI, genomic sequencing, cell-free DNA, and the treatments now are fascinating. So there is a report of a mother carrying a fetus with spinal muscular atrophy who got the drug risdiplam, and during her pregnancy, the baby came out with no evidence of SMA, was put on the drug, and is now 2 years old with no evidence of the condition. So very exciting. The fetal treatments now, as we learn to deliver genes and other small molecules and other therapeutics to the fetus, is going to be the next horizon and the challenge. And this gene therapy group associated with Inc., talking about gene therapies and cell-based therapies and biologics and small molecules and CRISPR and others. And there is this patient with a genetic disease at CHOP. It's normally lethal with hyperammonemia. I think it's carbamoyl phosphate deficiency. Okay, he's nodding. Good, I got that one right. And who actually, they took the genetic defect, sent it to a number of companies throughout the United States, and developed a CRISPR-like technology to treat the baby. And the baby is alive and well and eating better, gaining weight, thriving, and even during colds didn't get that hyperammonemia that normally you see. So a real challenge is going to be in rare disease, developing these technologies. As we make the diagnosis much more frequently, are we going to be able to use precision medicine approaches to treat these babies? So this is something that I'm really proud of, and it is the International Neonatal Consortium. And it was 10 years, as Kanwalji said, then a young lady by the name of Susie McCune approached me and said, we really need to do something about neonates. And I had participated in congressional hearings and things when we passed the 2012 FDA Safety and Innovation Act, really requiring more neonatologists. And I'm very, very proud of that Office of Pediatric Therapeutics and others, and have some wonderful colleagues with Lynn and Lily and Jerry and Melissa and Shannon and Ann and so many others in that Office of Pediatric Therapeutics have really helped drive the innovation for us and have been collaborators, and it really has helped the process move very much more quickly. And then we have some of our European colleagues, some of the regulators from PMDA who are here, and others that have really made a huge difference, and especially our parents. And I know it was wonderful for me to see Deb DeSenza, on every one of my grants, I write as the parent representative and get letters from her. It's really been quite helpful. So one of the things that we've really become interested in here is the use of real world data. And we were funded by the FDA and have now hit almost 400,000 records of health records, registry records, and other data sources like clinical trials. And it's really made a huge, huge difference. But I often say it was one of the hardest things I've ever had to do in my career is convince people to give us their de-identified data. It was a real, real challenge. And the sites really didn't want to share. If they did share, it's, well, you have to pay us for it. We're not going to just give it to you. So it really became a major challenge. But we put this real world data analytics platform together at CPATH. You can see all the sources on the left. I don't have time to go into it in more detail, but we de-identified the data. Cleaning the data was a real challenge. Those data sources were not the way they should be or were reported to be. But it really has made a difference. We're able to start looking at normal laboratory values. So this was just one data set in one group of babies, and you can see that we've developed an app to use this, and this is with platelets, and only babies 32 to 36 weeks gestation with the normal ranges. So that's something that's really made a huge impact. And we were able to take just one data set. So we cleaned one data set and we asked the question, does formula really contribute to the development of necrotizing enterocolitis? As you know, there are hundreds of lawsuits now filed throughout the US challenging this. And again, this was one small data set, and we're going to look at it in much larger numbers and expand the way we look at it. But you can see there was absolutely no correlation with the use of any formula exposure before 32 weeks post-menstrual age. So we're now putting as many grants as we can in to try to get the funding that we need to really make a difference. We've really enjoyed working with our FDA colleagues and meeting with them during the grant period almost monthly to help chart future directions. The real exciting thing for us is developing these models of digital twins and external controls from our data set. So instead of doing a randomized control trial with, say, 1,000 babies getting the drug, 1,000 babies getting the placebo, we are developing the techniques that only 100 of those babies will get placebo. The rest will come from the data set. And I hope this will work. And Lynn and I talk about it a lot. And I'm going to prove it to her. That's one of my missions in life, is to do that. And that's good. That keeps me going at my later stages of my career. But we're going to really see if we can pull this off to keep the controls just some to maintain the blind, but then to be able to vary the ethnic and racial backgrounds of the control groups, to do 1-to-1 controls, 1-to-2, 1-to-3, 1-to-4, etc., and really see if we can make a difference here. We're also going to be applying AI and machine learning techniques and really make a big difference. And this is what it looks like, and I'm not going to go through it, but this is our plan. For the future, to really use data-driven approaches, which is really what CPATH's expertise is in. I'm delighted to be working with them together and to really drive these innovation, these innovative ideas of moving forward. So more now than ever, there is an urgent need. Despite all our best efforts, things continue to crawl along, and I'm still waiting for these big success stories in neonatology to make a difference. The progress is being driven by legislation and regulations, and I again want to thank our global regulators for really working with us to help. And I want to thank our industry sponsors who who keep us afloat, keep these meetings going. And it really, I see that as the future in neonatology. I think the engagement of global collaborative networks, my colleague Mark Turner, who helped start Inc. with us from on the European side, is now running C4C in Europe full time. But I think these new diagnostic technologies, real world data, and novel therapeutics will drive innovation in this space. So I thank you all for working with us and being here today, and hope we have a really good meeting. Thanks so much.
Thank you, John. So, one of the things John very briefly mentioned in his talk was our real world data efforts, specifically focusing on addressing the lack of standardized lab value reference ranges in neonatology, which I think people who are either in neonatal drug development or working in clinical neonatology probably know that, when it comes to neonates, there is no normal. I mean, there's not going to be any 22-week normal premature baby anyways, right? But the fact is, we do not know how to define reference ranges for premature babies at all. For actually pretty much all reference ranges or lab values that are collected in clinical practice. So that's a really challenging issue. Because if you do not really cannot say with given degree of certainty what are normal quote unquote reference ranges for the most commonly used values, lab values in neonatology. You don't know what is abnormal. If you don't know what is abnormal, you cannot really use those by any given degree of confidence in your drug development programs or in clinical settings or wherever. So one of the things we wanted to change to do was to actually change that. In our real world data efforts, we are working on a really innovative effort where from the NICU records that we have, we extracted the labs, the most commonly used labs in NICU, and we basically standardized them, aggregated them, and created what we think are close to what is actually the reality, so that we can actually create generalizable reference ranges for these labs. In order to make our effort more accessible, we actually did not just stop at basically generating those reference ranges, but we have created a software application, a Shiny app, basically that gives everybody who wants to access to those labs. And one of the things our colleagues from quantitative medicine department, Nicholas Henshid and Rachel Zhu, they are actually going to provide a live demo of the tool that we have developed. And then we'll follow up with a panel discussion for the next half an hour, 45 minutes. But before we get started, I wanted to invite Nick and Rachel to the stage so they can actually provide an update on where the lab value modeling effort currently is, how the graphical user interface looks like, and then we'll basically see if you have any questions, and we'll answer those questions. So
All right, thanks. Hi. So by way of introduction, I'm Nick. I have a PhD in applied mathematics, and I studied precision cancer care. And I developed mathematical models for drug delivery in cancer. But since working at CPATH, I've pretty much gone across a spectrum of different disease areas, INC being one of the ones I've worked on for the last couple of years. And so the effort, like Kamaljeet said, is to try and take real world data and produce what we think is reasonable reference ranges for laboratory values. Okay. So I'm going to talk a little bit about the modeling behind this, a little bit about the data, and then we're going to give a little bit of background on what the graphical interface is going to look like, and give a live demo of that graphical interface. And then after CGIC, we're dealing with some final issues having to do with the authentication of accessing the app. And as soon as we get those issues worked out, then we'll give public access to get this user interface out into the world. So the data source, this is all from the INC real world data set that John talked about. And we specifically used three major NICUs for the data underlying this effort. And it ends up being about 16,000 subjects. And for the 14 labs that we are looking at, it amounts to about 1.5 million measurements. and that's specific measurement values that we have. And the 14 labs that we've looked at are listed here. These were put together by a discussion that we went back and forth in INC meetings. So if there's something missing from this list, we can certainly talk about it. We have basically any lab that you would find in the NICU setting, we have access to through the electronic health records, through the real world data. And so if there's something that you don't see on this list that you have a great interest in, please contact Convolgite, and we can get working on the next phase of this. Because now that the modeling pipeline is complete, it really should be relatively painless to develop new reference ranges for additional labs. And relatively painless. We also have, and this is a little bit having to do with the modeling. We're looking at 4 gestational age bins. So we have gestational age and postnatal age. We're binning gestational age into 4 different groups. Originally it was 3, and then we got feedback that we really wanted to see 4. And the plot that you see on the right is trying to visualize the amount of data that we have for each specific lab. Split into gestational age is the rows, and the postnatal age are the columns. And so the larger dots are the most number of measurements that we have. And in part, this is to show that one of the modeling challenges is that we have a lot of data for some subsets of this space, and we have a very small amount of data for other subsets of the space. So that creates challenges from the modeling perspective to account for small n problems. Okay, so the 2 main challenges that we had during the development of this modeling is, like Kamaljeet mentioned, there's not really a way to specify what a normal subpopulation is within this group of real world data. So in a sense, we have to approach this from a data-driven perspective. There's other things that we can talk about offline that we're planning to do as a way to enhance this. But for now, we're going to approach it just from a data-driven perspective. The mindset being that within this population of babies, there are, quote, non-pathological and pathological cases, and we're going to try and detect those purely from the data. And so that's what the plot on the left is showing, is this is a distribution of some lab value for some postnatal age, some gestational age bin. And it's showing that, as you can see, there is like a central peak that you can imagine is a normal group of subjects. And then there is a low and a high outliers or heavy tails, however you want to think about it. And the methodology that we selected was developed specifically for this problem of extracting reference ranges from real world data. And this method is called refine R, and I can provide a reference if you want, but if you just search for refine R, it's an R package that was developed at Roche. And it's specifically for this problem of extracting reference ranges from real world data. So we use that. And then the other problem that we have to deal with from a modeling perspective is longitudinality or covariate dependence. And so as people who work in this field know, the laboratory values change over time. And so the reference ranges have to change over time. And that's true in regular pediatrics, but it's particularly true in this population. And so just over the first 30 days postnatal, there's variability within the reference ranges. And so we have to deal with that as well. So this is a really quick overview of what the final modeling pipeline looks like. And this is, again, using work from this team at Roche that developed the refine R package. I've made a lot of improvements to this in order to deal with the data sets that we have. And hopefully, well, not hopefully, we will be publishing the methodological aspects of this at a later date. But basically, we use the refine R method to extract these ranges and then assign a non-pathological probability to every single data point. And that's what the color coding is on the far right. And when you look at the GUI, you'll see a color coding that has to do with the assigned non-pathological probability to each data point. And then we use a statistical method that's basically a regression method. It is a regression method that's called GAM-LSS, and that is what allows us to build the age-dependent centile curves. So the actual reference curves that you see on the right are built using this GAM-LSS technique. So, and I'm totally happy to answer any questions about the methodological stuff. There's a lot of technical details behind the scenes that we can get into offline if anyone is interested. I'll briefly talk about where we're going from here. I think that, as we mentioned before, there's additional things that we're going to try and do with AI and ML to specifically do patient-specific prediction. Again, my background is precision medicine, so I think very much in terms of right patient, right drug, right time, and developing something using this data set. I mean, it's such a rich data set that it would, we have to do something like this. It's definitely the future. And then we're also in the process of performing more complete validation and sanity checks against other aspects of what's in the EHR, like condition and observation. So I'm going to kick over to Rachel, who is our developer who developed the user interface. And she'll talk a little bit about that user interface and then hopefully show a live demo of that. Okay.
Thank you. So as you can see on the screen, this is a snapshot of the graphical user interface that we have. On the left side, in the little box, is where we have the set of inputs. So it might be a little bit difficult to see. Oh, awesome. So the first option you have is the list from the list of analysts you have. We have 14 of them. What we are showing here.
It won't work on the screen.
It's okay. We're gonna show a live demo later, hopefully. But here you can see AST has been selected, and in the second drop-down menu, you select a gestational age bin. Here it's extremely preterm. And once you have a combination of those two options, you will see on the right-hand side the curve plot. And currently, it's just it's also a little bit difficult to see, but you see the scatter plots on there as well, which is color-coded, and that is the individual patients or the records from the data. And if you would like to opt out of that, you just check the little tick box over there. Those are what that's for. And optionally, you can put in a numeric value that would be the number of days, the postnatal age. And what that does is essentially extracts a vertical line from the curve plot and display in a more straightforward way what the centiles would be for this selected lab and postnatal age. And the color coding correspond to the centile color coding in the curves.
Is that 2 standard deviation?
So we're using centile curves. So like the blue is median, and then it's 75th, 90th, 97.5. It is a potential future thing. If we could have that be an option if you wanted to see mean plus minus standard deviation instead of centiles.
I think in clinics, we're people use standard deviation.
I've seen this type of readout a lot in more of the laboratory science.
Want to see the frequency.
Sure. Yeah, if there's anything that you see here or that you don't see here and you would like to see here, absolutely let us know. And we'll have the live demo. We could also, well, again, like I said, we're still working through the authentication. So when we actually give the link out, then people can give feedback. But for now, I don't know if you want to just jump. How much time do we have? Do we want to just jump to the live demo, or
Yeah, we can do live demo.
okay, let's do that.
Is
Yeah, was there?
Is there a lot of smoothing that goes on with that model? Yeah,
LSS model is based on spline smoothing.
yeah.
It's a regression model, and so similar to any regression model, you have to pick a model there. So it is using spline smoothing.
Yeah.
And that's something that we encounter is if there's a lot of noise in the data, if there's a lot of irregularity, then the splines start to go crazy. So that's part of the model challenge.
Yep.
A really dumb question.
There's no such thing.
Yeah, I could do that.
So are you lumping all the values 22 to 27, or are they considered individually and then?
Right. So in the modeling that we're
Oh,
doing
that's right.
now, the gestational age is bid, and so it's pooled from 22 to believe it's 22 to 26. In a future version of the model that I'm currently working on, that will be treated as a covariate, meaning that it will be a continuous input value. And then you will have two continuous inputs, postnatal age and gestational age, and then you will have different reference curves for both of those continuous. But for now, and this has to do with the small n problem, the number of data points that we have, we pooled on gestational age. Because especially for the extremely preterm, in some cases, we would only have 10 subjects in a particular with a particular postnatal age with a particular gestational age. And it's just not enough to build a model out of.
And by the way, even though we're not allowed to give out access to the GUI just yet, but Nick and I will be here with our laptops. And I think we'll be allowed to show you if you're interested. Come up to us and we could pull that up for you.
Storing people's information and then giving access.
Yeah, and what we're gonna do today is distributing a link to log information from those who are interested so that we could follow up with you when that is available through the same email that you provide. So here's the actual tool. As we've discussed, you could choose from a list of drop-down of the labs, and then select a gestational age bin, and the GUI will pull up the corresponding curve plot for you. If you have, let's say you have a patient that is 15 days old, and what you could do is just go ahead and pull up the so that will correspond to this strip of value over here. And this plot is interactive, so you can zoom in, zoom out, and interact with it, essentially. Yeah. Yeah, you're allowed to do that as well. And then the second panel of the tool is also from the screenshots, is the refine our curves. Again, there's a drop-down menu for you to choose an age bin from. This is essentially binned a bit more widely than the previous page. And then, so those are the main functionalities of the tool currently. We do have a third tab that I would like to direct your attention to when you gain access to the GUI when it's available, to go to this link to provide us with your feedback so that we can make improvements that you would like to see in the future iterations of the tool. I think those are, yeah, so that's a preview of the tool. Yeah.
So, interesting question. That is a future improvement that we want to make. Part of that is, so there's two answers to that question. One, because Shiny is not a secure platform, I would say actually there's just one answer. Because Shiny is not a secure platform, we don't really want to do it that way, and I don't think you would either if you were, there's HIPAA, and even if it's de-identified. But there is a version of this app that will be deployed on our secure data platform. And in that case, you could upload your own data and then potentially run the analytics pipeline on your data to get these curves. I think that my modeling pipeline is now to the point where it could potentially be run in that type of setting where you bring your own data, you run the pipeline, you get these curves. That would, I think, be really powerful.
And that part of my question, part of it is how do you build them in? I mean, they were saying, 11% maybe were premature, but you guys have to build up that end with, people.
Well, right. So that's the other. Yeah, the other piece of this is we wanted to get started on this modeling as soon as possible, and so we took the first EHR record data sets that we had available and that were mapped by Will and his team to OMOP. We now have more. And so we now have a larger end that we can work with. That's definitely the next step, is to start incorporating some of those additional data sets that we've just over the last 6 months gotten ingested and mapped. That process is, and Will can speak to how laborious it is to do the
And not just integrating more data, even using some of the new cohorts. Independent validation.
Yes,
to
exactly.
show if
Yes.
the model works on a separate independent cohort.
Yes.
We can do that a few different
Yes.
times. This is really, I mean, every lab slip that we have, it's all the adult ones. People are using them on a routine basis. Don't understand. Plus, some of these things really, we want to help people learn. In other words, so a bilirubin of 5 on day 2 may be fine. Bilirubin of 5 on day 30 may be liver failure.
Right.
So, you know, how do we get people to understand the difference? Sugar and glucose, that is incredibly controversial, especially in the first day of life that people focus on. Can't even get agreement. Endocrine stuff.
Yeah.
Totally.
Even a term.
You know, terming.
Right.
Sure, absolutely.
Here's
Understood. Thanks. Yeah.
But there's another more mics here.
Okay. Yeah, I'd rather stand than hold. Yeah.
I assume you're collecting the data longitudinally, so a patient may contribute more than once or twice
Yes.
to the data set.
Yes.
On your first slide, you said you're also excluding patients who died.
Yes.
Is that completely excluding all of their data or not? Because I think,
Yeah.
see, I think that could be problematic because you may be able to want to use that information to predict
Yep.
outcomes in patients.
Yeah. Absolutely. Yeah.
So just a general comment.
Yeah. So, I mean, we can get into the weeds with that. Klaus is raising his hand. Why is Klaus raising his hand? Do you want to answer that? I always defer to Klaus.
The conversation that is happening right now is an expression of something that we live by at CPATH, and that is the learn and confirm paradigm. Especially with these types of tools are never done because there will always be more data, more questions, more things to work on. So the inclusion of the babies that died just calls for some form of joint models that also allow you to link how the lab values move over time linked to the time varying probability of the baby
Yes.
dying. And it's a fantastic additional evolution of the models.
Yes.
And then there comes the question that you raised about, okay, what's the context of use for the model? We started with one that was focused on ensuring that we started making life easier for drug development. But you raised a really good question about, yeah, this could also be used for clinical decision support. That's a different kind of animal with a different kind of evidence, but it doesn't mean that it's mutually exclusive. So this conversation is a fantastic expression of the learn and confirm paradigm.
Yes. Yeah. So, and just to bounce off of what Klaus is saying, you know, I treat this as an exploratory data analysis thing. And there's, once this is confirmed and has external validation, you can use this in the sense that everyone uses normal ranges for laboratory values. But this is exploratory data analysis that lets you look for additional questions like what is the time varying probability of some outcome, and how do you track not just single lab values but multivariate labs, because there's correlations between all of these. These are not independent variables, right? There's correlations between all of the different liver labs and the different kidney labs. And so developing something, and that's where the machine learning and potentially mechanistic modeling, and if we want to go way into the weeds, we could talk about, you know, actual mechanistic biological models of development in neonates and how those link to these different systems. And that's something that I think we could do with this type of data set, either building some of those models or validating some of those models. So there's a lot that could be done here.
Very interesting. Thank you for the presentation. So obviously, there's a variable degree of physician decisions and
Yes.
other diseases that impact these variables over time. So do you have the ability to, for example, identify a worried well baby, i.e. that is just developing and is not having an abundance of complications or an abundance of therapeutic interventions, because the spread is clearly reflecting that.
Yeah.
And so things that we've mentioned already, hematocrit, glucoses, which vary tremendously. And then if you look at the curves for things like AST and ALT, we're basically, unless you're in the liver failure category, as John outlined, or bilirubin, you're not, bilirubin is not a good example because you do things for that. So
Yeah, exactly.
how do you address that particular issue, which really defines
Yep.
what we're looking for?
Yeah. So to answer the question of can we identify that, if it's in the electronic health record, then we can identify it. It is a question of extracting that information. And that's where some of the new AI techniques with these large language models that we've heard a million things about, they can do that type of thing. They can start mining through electronic health records and looking for these specific queries like that. And then as far as like more, I would interpret as more of a statistical math person, the causal question of what is happening to the babies along their course. And the longitudinal, there was another question about the longitudinal course of individuals. Again, without getting too far into the weeds here, there are things that we can do to start looking at this not as just a pure data analysis, exploratory data analysis problem, but a modeling question of what is the variability among different individuals. And if you know modeling, you know about mixed effects modeling. That's what mixed effects modeling is really good at. What's the variability, inter-individual variability, and how can we model that with interventions? And the modeling gets very complicated very quickly. So I hope that's somewhat of an answer.
It would be great because I think most of these data are from US sites, and it probably will be important since we have so many records from children's hospitals to make sure that we get a mixture of hospitals, because the children's might be sort of skewed to a certain extent because you're getting the sickest kids being transferred there. So that's something to think about as we go through to make sure that we have other kinds of places, because they don't have delivery services, for instance. So they're just getting referrals of the sicker kids coming from outside. The other thing is to want to get data from our European and Japanese colleagues incorporated into this and see if there are geographic differences. But then many of these lab values have different units of measure. So how do we convert those over? I assume
That's easy to
with
do.
all these models, you should be able to convert them. And then to see again whether the European data matches the US, I think that would be absolutely fascinating to look at.
Yeah.
So, John, just a comment to this. We are actually on our way to incorporating additional international data. I think our next priority is going to be Japanese data, which we already have. Hide and our whole lot of Japanese collaborators have contributed the data to us. It is going to be incorporated next. That's basically what's going to happen next after that.
One more question.
Yeah.
The collection of the samples, this might be also quite different. You are going to electronic medical records in the hospitals, but the collection of the samples and the data itself might be very different. Has this been kind of unified somehow?
No, it's just an opportunistic sample collection, right? It is what it is. I think maybe that is something that can be incorporated and adjusted for in the modeling process.
Yes, and that's a really good question. I encourage you to follow up with Nick and Rachel, because that's something that even when we don't use real world data, even with using clinical trial data from industry, that is a thing. And a lot of times the most you can do is incorporate that as a source of variability into the model that you can't point to a specific covariate. And there are multiple ways of incorporating that mathematically, but that's a really important point. And sometimes you may end up with those, but the relevance of having those random effects that you can't know where the actual covariate lies are really key sources for additional questions for the learn and confirm paradigm, because you can then say, where is this coming from? How can we inform that? What additional information do we need to capture in order to figure out where the covariate actually is?
Okay, so this was really great. I just wanted to thank Nick and Rachel for providing the overview of the app and providing a live demo. So thank you, Rachel, for all of your work on this, and thanks, Nick, for the modeling part of it. So again, bottom line is this is not just a really an academic project. We, for the purposes of publication and publishing an article, we really do want people to actually use it in their daily, either clinical, day-to-day work or in drug development programs. That's basically really what our ultimate goal is. To that end, I just wanted to invite. We're going to have a very short panel discussion comprised of Nick Henshield, John Davis, Melissa Lestini, Carol Elegart, and Paula Balhata from Bayer, Belgium, FDA, Tufts, and CPATH. So, I'm just going to ask them to come to the stage so we can have a little bit of a discussion. Obviously, if you have any follow-up questions about the app or the modeling itself, please feel free to chime in, ask questions, uh, uh, and I'll basically try to be as quick as I can with this because we do want to make sure that we have enough time to answer audience questions as well. So some of these questions have already been addressed by Nick, but I'm actually going to start with Nick himself. Nick, I guess we already answered the question addressing the variability and levels across gestational and postnatal ages and across hospitals. But how do you think, or how quickly are we expecting the feedback that is going to be sent in by people, which we already are getting quite a bit, is going to incorporate it into the future versions? What are the most important future features that are you most excited to add to this version?
Just checking this microphone.
Yeah, I think it works.
So, I mean, to answer the first question of how quickly can features be incorporated, the answer is quite quickly, depending on how complex the feature is. As long as it doesn't involve reanalyzing data or doing the modeling over again, features can be included in the new version of the app, quite quickly, order of weeks. And then what was the second part of your question?
So I'll paraphrase that, right? So one of the things that I've heard a lot of people say is that they would like to, so they have a patient with a given set of criteria, characteristics, gestational age, postnatal age, a bunch of diagnoses, medications. They want to check basically, that at this given gestational age, given postnatal age, their AST value is whatever. How can they use, maybe not now or maybe in the future, how can they use this specific tool to place their baby inside that curve to exactly understand where that baby is, given all of the circumstances of the baby?
So this was touched on earlier, this idea of bring your own data. And I think that there is a version of this that would be amenable to be introduced into the Shiny app now, which is to have input boxes where you can say, this is the value that I want to place in this plot. And then the future version of that would be, I want to bring, I have a whole spreadsheet of data from a study that I'm doing, and I want to load that into this and see how they compare against the reference ranges. And so some version of that, whether it's in this currently deployed Shiny app or whether we have to wait for the analytics platform version of the app to be able to bring your own data, that's definitely a feature we're super excited about.
But I just wanted to actually ask Melissa and Carol, because one of the things that we debated is, do we put all comers into the lab value? So in other words, a baby with renal failure is going to have an elevated creatinine level. And we know that it's going to be 5 or 10 times higher than normal. Do we include that baby or do we drop them out? Do we take the wide ranges and drop all those out and just focus more in the middle because we want to focus on the normal values? So we debated that at length, and I'm curious as to what you both thought about that.
Yeah, I mean, I think it's a challenge, right? Because there's really no such thing as a healthy preterm infant, right? So finding that healthy infant is a challenge, and I think that understanding I think it was Victoria mentioned this, like, how do you know like what population you're really sampling? And I think that's an important consideration when thinking about this, because what it really depends on what you're planning on using this for, what what purpose? What is your intended use? And if your intended use is for it to be used as a normative reference index for preterm infants, then I would argue that you really do need to exclude those babies who have underlying pathology.
Right. And I have a follow-up question to Melissa regarding that, right? Because you were talking, I mean, from with your FDA regulator hat on, what sort of evidence would FDA need to consider this type of a tool as a trusted reference for maybe, protocol development, safety assessments, something that, if sponsors are using this type of a tool to report safety, what would it require for FDA to consider this tool to be, I guess, reliable and trustable?
Yeah, thanks, Kanwaljit. I have to say, when this tool was first introduced, I was like super excited from a clinical aspect because I was like, well, finally know what's a normal value for these kids, and we're not making all of these guesses about like, well, is it not? And then in terms of thinking about it from a regulatory perspective, there's really not one single guidance that addresses how FDA may look at this. I think FDA has published a series of guidances on use of real world data and real world evidence. And what comes through in all of those guidances, kind of the common thread, the common themes, are really that the devil's in the details, right? So understanding what your data source is, the quality of that data, understanding the population from which the data was culled, then also the curation process and cleaning of the data, what went into that, if there was data transformations that needed to happen and how that might have affected the data. And then what Nick started to talk about in some detail, the statistical analysis that's done and how the data is treated to try to get at your end result, what your intended use is for the data set. And then the clinical validation piece, making sure that what you have, your final product, is truly representative of what you think it should be representing.
Great. Thanks so much. Follow-up question to Paula. From an industry perspective, Paula, what do you think are the biggest barriers or biggest barrier to adopting standardized neonatal level reference ranges in your drug development programs?
Thanks. Well, FDA or the health authorities buying into and validation of those mechanisms, really. There's lots of things that we can discuss about how that will be incorporated into the databases as for the sponsors of the clinical trials. That will be that's technicalities that I can't go to, but that is something that I'm sure that can be incorporated in the clinical trials. There's from a technical perspective, but ultimately it ends up with acceptance of this data, mainly by the health authorities as well as from the community also. Yeah.
So this is a question to both John and to Carol. John from US perspective and Carol from EU perspective, how do you understand the reference ranges are currently established in neonatal intensive care units, and what sort of variability do you see across institutions? So maybe Carol, do you want to answer first?
Well, I have asked this in my institution and say, well, we just have observations previously collected in the patients and we report something like that as a range. But in international or multi-center trials, the major problem is that each lab has its own local aspect. And I shouldn't explain this for this audience, but we know that these lab values cannot be collected and sent to standardized systems because there's not much blood in the baby to do so. So there are some logistics who actually necessitate local assessment, and then the reference ranges are usually based on what the lab colleagues claim, irrespective of the sophisticated analysis. So this is, I really would like to say, this is major progress. You somehow provided a map, how to walk through the system. What I'm still struggling with, how will we use the map? From a safety point of view, will we indeed use standard deviations above mean, or will we use centile values? So that's something that I would like to think on from a safety perspective, to refer to the next session a little bit.
Yeah, I guess, no, that is certainly definitely true. But do you see variability across institutions?
Yes.
Yeah.
It's already alluded to. I just figured out last week, I think nobody knows, but liver values depend on how it is analyzed. Now, whether you add something, I forgot, sorry, but then you have 10-20% difference. And we had a discussion within European units, units located in Europe, and UK added something. And we did not, because it was too complicated. It was automated anyhow, so they didn't add anything. So the absolute values, even for liver values, will be dependent on the technique for the analysis. It's not rocket science. Assays will drive results. And it's very well known for creatinine. So yes, there is variability.
And John, from
And
US
that's why we want to do this and have enough units that we can incorporate that variability into the real world models, because it has to be real world, right? There's all different devices. As many of us clinicians know, that when I get electrolytes on my blood gas slip, that's whole blood electrolytes. And when I get it from the laboratory, that's plasma values or serum values, they're very different sometimes. So I think the more data we can incorporate into the system, the less variability there'll be in essence. But I think that's really the only way of doing it. And then I think taking the systems and letting everybody use it and getting that kind of feedback from the beta testers once we have enough data in there and can confirm some of the modelings. And I was saying to Nick, the best thing that comes out of this is to say to Nick and his team, try it both ways. Try it, as Melissa suggests, doing it with dropping out the extremes on both sides and really concentrate on the middle, which is where we want to define them, because you need to do it for safety reporting for trials and to know whether that value is normal or abnormal. And then having enough to integrate the different values. So for instance, ALT is specific for the liver, but AST may be specific for the heart, the liver, or blood. And so you need to be able to look at other values and even have the system log down, so you're in the 30th percentile for this, you're in the 50th percentile for that. You could take the entire Chem 20 in essence and give them the percentage breakdowns next to each value of where they are in the system. I think that would be incredibly useful. So then you're highlighting the ones that aren't normal, and then you can drill down into that a little bit more.
And I mean, that's an app development aspect. There's nothing about the data that says that we can't do that. That's an interface development issue. Can I grab a couple other points from what people said?
Go ahead.
So the two things I just want to mention. One, the statistical method that we're using to an extent does eliminate the outliers, because it is built to try and identify what the central peak is, and then it fits to that central peak. So it is narrowing in on what, again, in a data-driven sense, the normal range is. So there is some elimination. Now, what needs to happen is the post hoc verification of what subjects are being excluded and what is it about those subjects that is being excluded. Like and then that's where we get into the electronic health record and look at, okay, what's the observation? What's the condition table say? What do these different outcomes say about those subjects?
So John, just a follow-up question. In your hospital, Tufts, how are these reference ranges determined or decided, by the way? I mean, that's basically the crux of the question I wanted to
what we get on the laboratory slip are all adult values. So I don't even look at them. The only values that you can find are in the back of textbooks in the tables. So if you look at the Avery textbooks, 2 of them, you'll find in the back normal laboratory values with a few references that turn out to be small studies that people came up with. But that's all there is. So something like this is really an aggressive way of addressing these kinds of issues. I'm quite excited about it. And I gave a talk on drug development for a large conference in Florida a few months ago. And out of all my slides, and that was a 1-hour talk for 50 slides, that was the one that everyone focused on, that they were the most interested in, because this was a lot of clinicians in the audience who found that absolutely fascinating to be able to use a tool like this. So once we get a beta test version, I think the goal is to share it with people in this room and let people play with it and make suggestions for improvements and outcomes. And we have that. Same with Victoria with your BPD tool. I think there's opportunities to really use things like this to make a quantum leap forward, and I'm excited about it.
Right.
Yeah.
Yeah. Go ahead.
Fine. The other important aspect is how do you analyze the data? How do you conclude? What's the medical conclusion on the data? Because if we now have centiles like the growth charts, where do we draw the line in terms to determine, okay, this is a lab finding that is critical or this is not? I think that's also where we need to continue to work on, right?
We have done this, for instance, for creatinine. You have trend lines, and then you see suddenly that goes from P50 to P75, P90, and hey, guess the baby was treated with ibuprofen. So we can actually quantify these signals, and if there are sufficient clever people, you could again try to elaborate the links between an event or an intervention and a trend line over time. Exactly the same for liver, in my opinion.
Yeah.
So it can be done.
Can be done.
So Paula, from like industry perspective, if you're running a neonatal clinical trial or anybody else who's in the room from industry, how do you determine whether lab that you're collecting during the course of your clinical trial, whether it is within normal ranges? Like, where do you get those reference ranges from?
Most probably from the adults,
Uh-huh.
very honestly, and that's also why we have central labs usually in the clinical trials, because the methodology is so different from country, from site. That's the only way to uniform a little bit the conclusions is to get a central lab with all the caveats of it.
But
yeah.
It's basically kind of a black box at the end.
Exactly, it is. It is.
Yeah. So I was going to make the same comment. So in the adult space, drug development, it's relatively easy. You use central labs, so you can standardize and make sure that you don't introduce lab bias. In some of my larger trials that we've done where we don't use central labs, you obviously have the labs all sending their normal values, and somehow we try and integrate that into our database. But this becomes a real challenge, especially in neonatal patients, because the blood sampling you can't send off to central labs. There's so little of it. So we're going to have to essentially rely on each lab's method. So it does become a bit of a challenge, but it depends on what the use is. I think in terms of establishing normative values for general practice, this is a wonderful tool. But for clinical trial development, and especially when you're looking at patients with a specific disease, for example, I'm a nephrologist, so I'm obviously very focused on following serum creatinine, eGFR, and some of the other parameters. There it becomes a real challenge in terms of determining.
Yeah, but it may also be useful for you because you can take your patients and look at their creatinines and look at a response to your treatments and see that I think because of the fact that this is longitudinal, you'll be able to know, okay, well, you enter the trial when you're a week old and you're in it for the next 5 weeks. What are your values doing? And is that a response to normal variation that you would see, or actually is it a response to your treatment?
Yeah. And the one advantage is patients essentially serve as their own baseline also. And that's something we need to think about. In your modeling, you may want to think about that also.
I like to sell the message as we all use growth curves in our neonatal ward to see if the baby is growing. So why wouldn't we use lab values and see that creatine goes up or hopefully for you goes down as a reflection of improvement? And actually the absolute value can be the same, but still it's better compared to the trends over time. And it's not just central lab, it's also time-dependent physiology who makes babies interesting, but unfortunately a little bit more difficult.
And Carol's right, the Fenton growth curves are based on a few 100,000 patients from only 6 countries in the world. Yeah. And yet we use them in every country in the world too. And of course, there's always going to be variations, but it's the best we have. I look at the analogy that we're crawling now with this, and we still need to learn to walk and then run, as we use our developmental assessment. Yeah, but I think this is where we're making some really good progress here.
So to Carol, what do you think are the most complicated or important challenges that you believe would be pertinent for us to consider in terms of harmonizing these lab value references internationally when we are trying to potentially incorporate Japanese or European data into what is basically right now predominantly only US based?
Yeah, I think there's for sure value to add other continents or other type of units, as you refer to. But sometimes you have to agree on not the best potential solution and agree that, as you said, Fenton growth curves are not based on a million, although we have perhaps billion data, they're not based on that many data. So I strongly believe that the central type of concept would be very informative, even at the individual level. How does it evolve over time in my intervention or randomization? So that's something that I strongly believe in. And secondly, as I already said, from an adverse event type of assessment or adverse drug reaction type of assessment, we will have to think on how we will use this tool. And together we know hopefully more than all of us individual. I would be inclined also to consider centile values instead of median plus 2 or 3 or 4, whatever standard deviations based on the normal values. And this way you would detect your basal kidney failure. So I really would like to urge also to already think on how will we use this tool beyond the development, which is highly appreciated, but we have to think on how will we implement this in the adverse events assessment and in clinical care.
Right.
These are the two issues.
So yeah, go ahead.
Sorry again for jumping in, but again, coming.
Yeah, I think so.
So just coming, I was thinking about this issue about scrubbing, for outliers and things like that. In some sense, I say you have to almost essentially do it to establish your normal values. But at the same time, in drug development, it's something that I don't think you can ignore because there may be certain signals that if you're going to start throwing out outliers, you may just be missing something. And I remember as a young gentleman at Merck, when I first started speaking to our statisticians, they used to chastise us when we went to throw out data because they said the shuttle disaster was because people threw out the O-ring data so we have to really think about throwing out. And the advantage now with the evolution of AI and us being able to perhaps use EMR data and actually collecting other parameters that might be associated with outliers is something on the horizon. So we just need to think that we will eventually have the ability and tools to actually discriminate.
I think it's going to be doing it both ways. And I'd be curious if Lynn or Jerry or others have, because it's really going to be driven by the regulators to say this is a system we can accept and this is one we can't. But I think doing it both ways and having the option, I don't know if it'll be possible to switch back and forth, but I think looking at it, I don't know if any of you folks or Susie even, if anyone has any thoughts about this.
Well, and as she's going to Lynn, I'll just hop in because one of the things I was considering as I thought about the talk today was really not those babies early in life that we get a lot of labs on, but those babies later in life, probably where Nick, you were struggling with getting data points. And when we get those labs, it's oftentimes because we're worried that there's something wrong. And so my concern about, I think looking at it both ways is a good idea to understand the differences between the data, because my concern is that you're going to get more pathologic results later on.
Right. So if you're on diuretics, we're going to follow your electrolytes. Whereas if you're not, you're just a grower and we may not get any of those. Yeah.
This refers a little bit to the caliper discussion we had a few years ago. You don't have normal babies in NICUs. They should be home.
Which the point I would make there is that, and I've been thinking about this for a couple of years now, about it seems like we're trying to fit I wouldn't even say square peg round hole. It's like a very oddly shaped fractal peg in another fractal hole, but whatever. I mean, like the concept of normal ranges just almost doesn't apply. So it very much will, and I hope that we find a context of use that makes sense for this analysis, because it was a lot of work. And if there are other approaches that will be more appropriate, that are more along the lines of a precision medicine approach, where I want to know what's happening with this baby specifically, that's where other modeling approaches and AI and so forth, I very much hope will apply to this, and could answer some of these much more complex questions.
I think
This is a simple approach.
I think Glenn, you have a comment? Go ahead.
Yeah, so I've been sitting back here and listening with great interest because I was asking Susie, I said, the first question I had was, so this is great, but what are you going to use it for?
Yeah.
And I think that what we should not do is wait until we figure out the perfect use and just sit on it until we do. I think what I'm hearing right now is, for the purposes of understanding adverse events in laboratory values, that what has been created is this normative range. And yes, it's imperfect because all of the newborns, all of the premature infants that you have included are sick for some reason, but they're hopefully sick in similar ways that give us normative ranges. So I think the first step is to acknowledge that there are some areas that we have uncertainties, but to also say, have we ever had normative ranges before, even close to what is being presented here now? I mean, by gestational age and, longitudinally, this is, very different. And I think if we limit it to certain contexts of use, like we're not going to use this necessarily as the efficacy endpoint, as a biomarker for a, accelerated approval for something. But it seems to me like you have developed for the adverse event reporting. This seems to fit very nicely. And look, it's out of range. It's not, and compared to what we're using now, which is an adult range, seems again like it's much, much better. And then I think to move forward, that associated data, the data that describes this is a child who had, kidney failure or was on diuretics or heart, post-op heart, that as you accumulate these large amounts of data and use, I think as Nick is saying, these, large models, AI to hone in, we may end up having different categories of laboratory values for different clinical situations. I think maybe that's the next step.
Yes.
But I do see a real use right now, currently, improving on what we have.
Yeah.
And I do see amazing applications in the future.
From a clinical standpoint, I mean, we have nothing. So this is a major leap
Yeah.
forward. I agree with you, Lynn. Just to be able to put that on an app in my computer in the NICU and let any nurse practitioner or PA or physician go and look to see, I think that would be a major step forward for us.
I think there's one more question. Did you say? Oh, okay. So unless you have from the audience any more questions, we will take a, yeah.
Just a 30-second leap off of that. This is something we can incorporate in our feedback form, because similar to what Klaus was saying with the learn and confirm. I mean, we need the feedback from you to understand what is working and what is not with this. And so when we distribute this, that's why we added a link to a feedback form, and then we'll find a way to compile that and try and start a conversation so that we can get everyone in the same room and saying, I'm seeing this happening with these ranges, or I'm seeing this use case, or I'm seeing this potential improvement, and this is where we can go from that. So compiling all that information from this audience is going to be super powerful. So
And getting that one data set large enough that you have, has been alluded to, enough patients on, especially the outer ranges, as Melissa describes appropriately, to be able to have confidence that those data points are valid, I think will be equally as useful. And then when you also say, well, we've used 2 other cohorts, and they really do seem to fit, those normal ranges seem to be okay, then I think you turn that out to us and let us start really playing with it.
Yeah. All right. If no, yeah, go ahead.
So during break, you guys want to network, we do, but if you don't mind going maybe out and board elevators to the next meeting.
Yeah, just to, yeah,
Oh, no.
good idea. Thank you. Uh,
You got a
so,
fucking point.
okay, well, that was a great discussion. Thank you. Ultimately, as I said, we did not do this as an academic project where our end goal is to write a manuscript and publish a manuscript. We really want this tool to be used actively by everybody, whether in clinical setting or in industry setting or in regulatory setting, wherever it might be. And that's really the goal for this entire discussion. In the next few days, we will be releasing this tool for public testing. I think there's a link here somewhere which we will be forwarding to you all that basically allows you to register, put your information in, and then you will get access to the tool within a few days. So that's the process behind that. And we will take a short coffee break and be back here at sharply 10:30 a.m. for the next session. Thank you. Thanks, everybody.